Well, we're going to move straight into our talk by Samantha Smith. She is our research tech here, and she's been with us for about a year. And while she's instrumental in so many different projects, whether it's flora or fauna, she's really taking a keen liking to understanding bivalve abundance and health in the Indian River Lagoon. So specifically, she'll be talking today on oyster spat recruitment in the St. Lucie Estuary and then IRL.
Hello, everybody. I'm just going to go ahead and put this in presentation mode. Does it look okay? I hope so. Welcome. As you just heard from Lorraine, my name is Sam. I'm a research technician here at FOS, and I'm going to talk to you a little bit about our new monitoring efforts as far as oysters are concerned within the St. Lucie Estuary and southern Indian River Lagoon. I hope that you enjoy it. A little bit of background for individuals that are not familiar with this area. The St. Lucie Estuary and Indian River Lagoon are also fed freshwater inflows from Lake Okeechobee, and that's the central lake that you can see in the image there. It has outflows to the east and west. On the western side, you have the Caloosahatchee River, and on the eastern side, we have the St. Lucie River. The portion of the waterway that we're most concerned with is the southern Indian River Lagoon and the St. Lucie Estuary, as previously mentioned. We are positioned optimally to study those areas, especially at FOS, and they are considered some of the more impacted waterways, especially as it's related to land use and watershed management practices. So the Indian River Lagoon stretches 156 miles from the Ponce de Leon Inlet down to the Jupiter Inlet in Palm Beach County. And it's previously been coined the most biologically diverse waterway in North America, expected to house roughly 4,000 plants and animal species. So, with that being said, the body of water that we're most concerned with is the St. Lucie Estuary. The St. Lucie Estuary is fed by 3 different tributary canals. You have the C44, the C23, and the C24 canal. C44 contributes water directly from Lake Okeechobee, and the C23 and C24 canals contribute water from land management practices such as agricultural uses or even residential runoff. With that being said, a lot of fresh water flowing into a system can really impact and influence some ecosystem shifts. What we're concerned with as you look into the St. Lucie Estuary, it's broken down into 3 different areas. You have the central estuary, the northern fork, and the southern fork. The southern fork is most exposed to some of those freshwater inflows as it's closest in proximity to the C44 canal. And as you go through that system, that water flows then out into the Indian River Lagoon and farther out into the ocean through the St. Lucie Inlet. What does that all mean? We're really concerned with any of the ecosystem shifts that might be taking place as a result of some of those freshwater inflows. If inflows occur too often or at a rate that is too fast for the system to adapt, of course the shifts might be a little bit more adverse or organisms aren't necessarily able to come back up to speed. One of those organisms being the eastern oyster, Crassostrea virginica, which we will focus on in our monitoring efforts. So what is an oyster? The eastern oyster, Crassostrea virginica, is a filter-feeding bivalve. Under optimal conditions, they can consume up to, or filter up to 50 gallons of water per day, and they consume phytoplankton and algae from within the water column. Oysters are able to act as bioindicators, and the reason for that is that anything that they are consuming from their environment or the functions that they are performing directly indicate the overall health of the environment that they are situated in. So why are oysters important? They provide essential ecosystem services such as habitats and nurseries for a wide variety of plants and animals. They act as a food source. They participate in nutrient uptake, sequestration, and filtration. Not only are they taking in algae and other things from in the water column like their phytoplankton that they would normally feed on, but they're also able to remove things like heavy metals or maybe other chemicals and either sequester those things in their tissues or secrete them in feces or pseudofeces. They help reduce sediment resuspension. So as sediments are suspended in a water column, they're able to settle on oyster reefs and actually remain in that area without reef suspending. And they help with shoreline erosion prevention. As you have more reefs built along the shoreline, they're able to reduce wave action and actually prevent that erosion. There are additional benefits, of course. So there is a very large commercial industry dedicated completely to farming oysters. There's a large economic component that's associated with that as well. There's also recreational fishery stock improvements that can come out of putting oyster reefs in place. So you'll have return of benthic species or other types of sport fish that are very popular in this area, like sheep's head and snook and things of that such. And then lastly, the scientific interest in understanding how these organisms function and what their physiological processes are like in relation to the environment that they're living in. So an oyster's life cycle. Oysters produce sexually, so they release sperm and eggs into the water column where fertilization takes place. And from roughly 14 to 21 days, they are free-swimming larvae that have the opportunity to settle on hard substrate. Now, if larvae do not find hard substrate within those 14 to 21 days, they will die. And they are actually brought to different types of substrates, whether it's reefs, pilings, docks, things like that, by chemical cues from adult oysters. As you can see on the right-hand side there, these are actual spat and what they look like on some of the stringer shells that we've collected over the last several months. It's really interesting to see, and I think they're kind of cool looking. Stressors to oysters. Oysters do not perform well under certain conditions. So if a body of water has a salinity that's too high or low, oysters will stop performing their essential function. Additionally, if temperature changes, so any temperature that drops below 68 degrees Fahrenheit, oysters will not spawn. They actually enter a dormant period. And then anything above about 30 to 33 degrees Celsius, oysters experience heat stress. So that's not great for trying to spawn, grow new oysters, build reefs, things like that. Dissolved oxygen is the amount of oxygen available to organisms in a body of water. So of course, if there's not enough oxygen, respiration won't take place and animals will die. Predation is a natural cycle, of course, but organisms, especially invasive species, if they get involved, can really be detrimental to some of these reefs. Low population densities also impact the success of an oyster reef. So if there are not enough healthy adult individuals, new spat do not know how to find these healthy adults and begin settling and growing. Those chemical cues are not being emitted at maybe an abundance or a frequency that the spat are able to pick up on, so they don't settle and the reefs won't grow. Sediment accretion and burial. If sediment is being deposited onto a reef at a rate in which the reef is not able to outgrow it, the reef will eventually suffocate and die. And again, there are things like disease and parasitic infestation. So dermo is a very common disease that we hear about, especially in warmer waters, or Perkinsus marinus, and parasitic infections like boring sponge. They can actually result in really large die-offs on reefs. The objectives of our monitoring efforts. So we aim to identify changes in the environment as it's related to spat recruitment and overall reef health, and hopefully this information will help us better understand the condition of the southern Indian River Lagoon and the St. Lucie Estuary in an effort to guide future restoration efforts. You see some images of us out there. Marco's up in the corner doing some spat counts. Site selection. We chose 6 restored reefs within the Indian River Lagoon and St. Lucie Estuary. 3 of those locations are in the IRL, and 3 of those locations are in the St. Lucie Estuary, isolated to the southern and central estuaries. Unfortunately, in the northern estuary, consistently high bacterial counts make it very difficult to conduct monthly monitoring efforts. So each month we go out to these sites and we collect situational environmental data or data that we collect at the time of sampling. We're looking for temperatures, salinities, pH, dissolved oxygen saturations and content, as well as tidal information. Each month we also look at the individual spat recruitment at each site for each oyster tea that's put out, but we'll talk about the teas in a little bit. Biannually, oyster abundance, percentage of live and dead reef cover, and percent coverage of mussels and algae are all visually quantified. And this is to get sort of an idea of population density. We also look at the total reef area, and we use collect biomass data. So methods in collecting our environmental parameters. Environmental data is collected for cross-comparison with the spat counts, biomass data, and population density data to hopefully paint a better overall picture of each individual reef. And this data is collected using metered devices like YSI instruments, GPSs, thermometers, meter sticks, all standard stuff. Population density is taken biannually, and it's conducted using half-meter squared quadrats. They are randomly placed along the entire reef area at a site. The percentage of live and dead oyster reef coverage is visually quantified. The substrate type is recorded, whether or not it has a sand bottom or a muck bottom, and then also the percent coverage of algae and mussels is visually quantified. You can see a few images from some of the ground truthing efforts, as well as some of the actual biannual sampling here on this slide. Oyster tea construction. So we set teas out in order to compare the spat recruitment to the population density, and we do this using oyster teas. The oyster teas are constructed using 3/4-inch PVC cross-arm design that is actually outfitted with an oyster stringer. So a stringer consists of 7 clean, dried oyster shells with a centrally drilled hole so that a 14-gauge galvanized steel wire may be strung through the shells. A loop is added at the top and the bottom of the shells themselves to prevent loss, and then the stringer is fitted onto the oyster tea. Additionally, and this was to address a methods question, 4 oyster teas are deployed at each site. 2 of the teas are outfitted with a double stringer design, so each arm receives a stringer, and 2 of the teas are outfitted with 1 stringer and lead weight balancer. Oyster tea placement. Oyster teas are deployed every 4 to 5 weeks, so on monthly intervals, and at optimal depths to monitor spat recruitment. They're deployed within the same depth range for each site and at the same GPS point monthly. The teas are not only placed within an optimal depth range, they're also placed at least 2 to 3 meters away from each other and an additional 2 meters away from mean low tide at each site, adjacent to a restored reef. The teas are set allowing stringers to hang at the depth that corresponds with the greatest visual density of live oysters on the adjacent reef. The idea being that those live oysters are sending out chemical cues to spat, and that is ideally where they would intend to settle. Here's the individual oyster tea orientations by site. You can see under some of these images some reef history, relative number of reef with rough size estimates. You can also see their actual linear array orientation. Note at the middle, at the bottom of these images, you'll see the Hogs Cove site. Now these teas are oriented in a different spatial form, and this is at the request of the homeowner. This is a private residence. Methods for our spat count. Oyster tea stringers, of course, are collected every 4 to 5 weeks with the redeployment of the teas themselves. They're placed in a seawater flow-through tank system until they're processed. At processing, individual spat are counted and recorded as either live or dead. We also go ahead and quantify any of the epibiota on the shells themselves. So you'll notice in some of those photos that there are tube worms, encrusting bryozoans, different types of algae, so that we're not only just looking at spat, we're also trying to see what else is coming to that area and finding usable habitat. In this graph, you'll see the spat recruitment by site. So this breaks it down to each individual site. Notice that this is not necessarily over time, this is in total. On your y-axis, you have the total spat in numbers, and at the bottom you have the sites. What's important to note here is that we are seeing significantly different levels of spat settlement at each site. The darker figures on this graph are out of the Indian River Lagoon, and the lighter figures on this graph are from the St. Lucie Estuary. Now, as I said in the beginning, this has really only, these methods have only been ongoing for about 12 weeks, so our data sets are somewhat smaller, but it's still really interesting to see that we have such a difference in spat settlement amongst the sites. However, it's also nice to note, too, but there is a significantly similar amount of spat settling in the central estuary at the private residency as well as at the reference site. Here you can see our monthly sampling results as they're related to spat recruitment per shell. This breaks it down even further into the Indian River Lagoon and the St. Lucie Estuary. What's important to note here is that we are, again, seeing significantly more spat settling in the Indian River Lagoon, or the southern portion of it, than we are in the St. Lucie Estuary. Salinity variation. As we said in the beginning, oysters do not prefer salinities that are too high or too low. So here it's really interesting to see that in areas of high salinity, where you have this on your y-axis, over time, we are noticing that there's less of a gradient and more of areas of high and low. And it's also interesting to note in comparison with the last graph that those areas of high salinity are also experiencing greater spat recruitment. Again, this is all very preliminary data, but here you can see a comparison in the number of spat settling per shell, as well as the different types of epibiota and fouling organisms. I would like to note here that so far we have only quantified tube worms and leeches, but going forward we would like to expand it to include mussels, surf clams, different types of algae and such. Oyster processing. So the next set of processing related to determining the overall health of the reef is to actually look at the biometrics for each reef. They may provide indications of overall reef health and success, and that information will help us then guide future restoration efforts, possibly provide commercial industry improvement advice, and actual information related to the overall health of the St. Lucie Estuary and Indian River Lagoon. With that being said, we'll go into this. Oyster processing occurs biannually. 10 adult Crassostrea virginica oysters are harvested from each monitoring site. They are randomly sampled off of the total available reef area. They are transported on ice and brought back to the center, and then refrigerated into process. The data collected includes the shell height, lengths, and widths of those individuals, a condition index, as well as physiological condition, whether they are fat, medium, or watery in condition, and sex ratio. Oyster processing, the condition index. In this image, take your time and look at it. It's actually kind of neat. So a condition index is a formula for grading the overall condition of an organism. And the measurement allows researchers to evaluate how much energy an organism has devoted to its growth and development. And it also may pinpoint environmental factors that are either prohibiting its growth and physiology or physiological condition, as well as its function. So here you can see an oyster that looks watery and in relatively poor physiological condition, but it still has a partially visible gonad. So this is kind of neat to see as we go through. So oyster processing for sex ratios. Gonadal development. In order to see whether or not the gonad is developed, you actually have to do a dissection, open the oyster up, and locate the gonad. If that gonad is visible, a scraping is performed, and the gametes are analyzed using a dissecting scope. Here in these photos, you can actually see images of what female oyster's eggs would look like. Quick shout out to Tom and Nicole. Thank you for the equipment. The presence of oocytes or eggs would indicate female oysters, and the presence of spermatozoa or sperm would indicate male oysters. This ratio can provide a representative figure for how many potential adult female and male oysters are on a reef. So what's next? We would like to take all of this information as the project continues and actually run appropriate statistics and analyze any of the trends. The sampling is set to continue year-round, with the biannual sampling occurring again in spring. And the outcomes are, of course, for use in planning future restoration projects. So thank you for attending. It was a little quick, but I would like to open it up for questions, and also thank our research department and our development department for putting all of this together. My email is here as well. If there's anyone that has a more detailed question that does not want to ask in the chat box, please feel free to email me. So I will take it over to Q&A. All right, so hang on one second. First question is, have you seen an increase in macroalgae in areas that could be potentially spat settling areas? Is that a concern in the IRL? I can't say anything with any sort of statistical significance at the moment, but anecdotally, what I am seeing is that, especially in certain seasons, there are greater amounts of macroalgae, such as brown or green filamentous algae, settling on these shells, but I can't say anything definitively. That would be a concern, but I'm not exactly sure what would cause an increase in macroalgae. So that's like a multi-layered process, and probably would lead into another study at some point. So thank you for the question. Savannah Lacey says, how do the larvae move toward the substrate, which the adults are leading them toward through the chemical cues? Do they physically swim? And how much control over the mobility do they have? How much are they at the mercy of tides and flows? These aren't really, really small organisms or larvae that we're talking about. They're microscopic, and I'm certain that a lot of us know that, but they do freely swim. They have, I believe it's cilia. I can go back to the diagram, but they have an ability to swim. Whether or not they're strong swimmers, I can't particularly say. They are really, really small, so I'm sure that they are at the mercy of tides and flows, and I would be really interested to see what the flows are like in the St. Lucie Estuary down the line. Was there an observed difference in the size of the oyster reefs / new recruitments in areas with routinely high current where there may be more food? Anissa, I'm not really able to say that because I haven't actually analyzed any of the currents. Right now I'm just trying to gauge whether or not these reefs are recruiting spat, and as far as the reef itself is concerned, the overall health as is. I don't know what's affecting it at the moment or how it's going to look down the line, so I'm really excited to see where it goes. Next we have, can you explain what might be causing the large error bars in the graph of spat recruitment by site? Yes, actually. So that actually has to do with whether or not stringers are lost in the process of sampling. So while we have put these new methods in place to minimize loss, corrosion is still really, really, from a design implementation standpoint, problem. So you're using galvanized steel, and it's only good, especially in marine conditions, for a certain amount of time. So in the beginning, especially as we were ironing out what we wanted to do with this study, it was difficult to determine what method and what materials were going to work best. So some of those losses can contribute to some of the higher errors that you're seeing there. Can you show the pH readings during the trial? Absolutely. We're still compiling everything and putting everything together. So we have lots of different readings for DO, pH, salinity, temperatures, you name it. And right now it's just a matter of trying to really narrow down exactly what it is that we would like to show correlations between, if there are any. Why do you use lead weights on some of the oyster teas rather than a second hanger? Thank you, Tom. What a wonderful question. I was hoping somebody might ask. So this is a methodological question. I would like to ultimately reduce the amount of time, effort, and cost associated with materials in implementing some of these restoration efforts or monitoring efforts. So the thought behind that is that an oyster tea with a double set of stringers is hopefully going to show the same statistically significant evidence of spat recruitment as an oyster tea with a single stringer. That would then allow, especially smaller organizations, to implement monitoring projects and possibly cover a larger area using less material. So far, and from what we've seen out of deploying those two different types of designs, there's actually no significant difference in spat settlement on a single T as opposed to a double T, and that's with a p-value of 0.8049. So I hope that answers your question, and I'm really, really looking forward to seeing how that turns out as well. One second, I gotta scroll. 1, are spat counted from the bottom and top of the shell? And 2, are they averaged per site? Yes, spat are counted on the top and bottom of each shell. However, for a stringer of 7 shells, the topmost and bottommost shell are discarded, so that, or not discarded, they are looked at, but the data is not factored into the overall statistical analyses, and this is so that each shell receives the same treatment. Are they averaged per site? They are, in a different set of graphics that I do not have prepared yet. I'm still in, 12 weeks of infancy here. And 3, are you standardizing the numbers of days as, I'm sorry, I'm not understanding that part. Barbara, are you standardizing the number of days as days they are dispersed vary? Barbara, if you can rephrase that for me, unfortunately I'm just not quite understanding it. I'm really sorry about that. Vincent. Hi, Samantha, could you explain the lead balancing weight treatment? What is the reason for that? As I previously explained with Tom, it's a methodological question. As far as the lead balancing weight, it's so that the T maintains the same depth within the substrate. The lead is basically there to keep it balanced and keep it from tipping in wave action and things like that. What are the invasive predators that you're finding on oyster reefs? Actually, Zach, I haven't found any invasive predators just yet. Recently, in doing some of our biomass assessments, as we just started them this week, we did see some pea crabs inside a few of the oysters, all of which located in the Indian River Lagoon. I did not find pea crabs in any of the oysters dissected from the St. Lucie Estuary, but again, that's all anecdotal, and I don't actually have any concrete data for those numbers. Is there a baseline number of spat to maintain an existing reef? Felipe, great question. I actually don't know the answer to that. If anybody does know the answer, would you mind popping it down in the Q&A? It would be really interesting to know. I'm still referencing some papers and some other monitoring efforts from partnership agencies in this area, so I'm not quite sure on that one. Anissa, are there any factors that influence oyster sex like temperature, or is it just random? Oysters start out male and they eventually transition into females later in their life, or they have the option to do so. I'm not quite sure whether or not temperature, salinity, or things like that affect at what rate that process occurs, but spawning in particular will not take place at temperatures lower than 68 degrees Fahrenheit. Susan, are you looking at the presence of microplastics in the oysters? And if so, what are you finding? I'm actually not, Susan. There was a study conducted by the University of Central Florida out of Dr. Linda Walters' lab with the master's holding person, Casey Craig. She actually did a study looking at whether or not oysters were processing microplastics and actually retaining them in their tissues or excreting them in feces or pseudofeces. And she actually had some really interesting findings. If I can find a link, I will pop it down in the box. Soundscape ecology has demonstrated significant effects on the ability for spat to settle. As a part of your situational environmental parameters, are you documenting boat traffic or noise at the time of sampling? No, I actually haven't looked into any noise pollution, but it's a really good point. I'm not quite sure what kind of equipment we have available to us to conduct something like that, but that's also really interesting. I'm looking forward to the questions that will come out of some of this data. I think it would be really great to kind of go into some other studies in this area, especially as far south as we are. Is the difference in spat recruitment an indication of the number of spat in the lagoon and estuary locations, or differences in how they are attracted to the existing shells? I don't know if it's indicative of the number of spat in the lagoon. I would actually need to look at different types of changes in spat fall over time, and as it is right now, I don't have a large enough data set to do so. It would be really, really interesting to see. That's kind of what this is for. We want to set a baseline for exactly where spat are settling and how they're doing as they continue to grow and develop. Differences in how they're attracted to the existing shells. Again, the only thing that I know definitively is that adult oysters emit chemical cues. Whether or not they're attracted by anything else, I can't say. Did you end up comparing strings with and without metal weight? Not T's in particular, but yes, the double stringer method versus the single stringer method. And again, with that p-value of 0.8049. There was no significant difference in the amount of spat settling on a single stringer T versus a double stringer T. Do we have ways of quantifying spawning? Have you compared spawning to spat settlement to determine if abundance or lack of spat settlement is related to quality of spawn? I have not done that yet, and I'm honestly not sure. Again, I'm really looking forward to diving into a lot of these questions. Jerry Snyder. Hi, Jerry. Are you finding many mussels at the sample sites? That's actually a really great question. In the St. Lucie Estuary, at least anecdotally, again, I don't have concrete numbers for this, I am seeing a lot of mussels. They are ribbed mussels. I haven't looked for invasive mussels, going back to kind of that question about invasive species or even space competition. But yes, Jerry, I have seen mussels at some of the sample sites, mostly in the St. Lucie Estuary. Are these organisms as good as the oysters at filtering? I don't know, and I actually haven't come across a lot of studies where individuals work with mussels in this respect. That's another avenue that we can explore down the road. How much do these reef sites improve carbon sequestration over a bare bottom? Again, an excellent question. Off of this, we would really like to look into how nutrient cycling takes place at some of these locations. Thank you for the complimentary. Did you look at settlement rates related to density of reef sites? No, I did not. If it was not presented here, then it hasn't been looked at just yet. Again, we're only in week 12, but thank you for the question, and it's honestly a decent suggestion. Will the monitoring project catch the major spawns in the estuary during the year? Yes, Mark, it is an annual project, and we hope to notice the major spawns for the estuary over the course of the year. I'm not sure when those peaks will take place. I know that there are literature references that indicate when spawning should be occurring, but I'm interested to see if our area responds a little differently. Thanks for answering that question on the lead weight treatment. I agree, Vincent. I think it would be a great way to cut down on processing time as well as materials cost. Barbara Welch, standardizing the number of days the stringers are out 30 versus 28 in the average. If the hangers are out longer, they could have more spat versus a shorter period of time. Oh, I'm sorry, Barbara. Okay, I see what you're saying now. Yes, they are standardized. They go out between 4 and 5 weeks. They do not go out longer. They do not go out shorter. And that is every single month. So we've been able to maintain that schedule so that we can narrow down, at least within a certain range, yes, the amount of spat that are settling over that period of time. The answer, Vincent says, the answer to the minimum spat needed to maintain reef is, he believes, unknown. He was in a meeting on the importance of shell budgets for a reef and how much shell is needed to sustain a reef, either through oyster recruitment, growth, and death, or culture supplementation, and it sounds as though that figure is unknown. Susan Hamburger says she was part of Casey's microplastics study, collecting water samples, filtering and such, and is now collecting samples at outflow pipes in the Indian River Lagoon. She was wondering if FOS was including microplastics in oysters in our project. We are not currently. Depending on how this goes and progresses, we may be able to in the future, but no, not at the moment. Of course, I mean, as a nonprofit organization, we're always interested in collaboration. So if at any point Casey or anyone else would need assistance or any other projects for collecting samples to that respect, we would be very interested. And then last but not least, we have a couple more comments from Vincent. He says that we compared mussel filtration to oysters in a project with the Smithsonian. Oysters do filter more. I suppose that could be expected depending on size differences, but they were more finicky in those experiences, in those experiments. And I could definitely see that. All of the different things that influence an oyster's overall health and development that I'm noticing, they just appear to be a little bit more fussy. The PIs from the Smithsonian did publish it, and he said he would double check. And lastly, again from Vincent, we have shell budgets are being modeled in Gulf states and will be applied to Apalachicola for fisheries interests. So very, very interesting information. And this is what we were hoping to get out of this. I cannot wait to explore all of the sort of auxiliary questions that come off of a monitoring effort like this. So I thank you all for attending. And I really, really appreciate everybody that helped put this together. So I will give it back to Lorraine to close us out, and thanks again.
Wonderful. Thank you so much, everyone, for attending, and thanks for all the amazing questions that you guys had. Like Sam mentioned, part of having this conversation about our 12-week projects was just to start the conversation of where we're going next, what we're up to, and hopefully these questions turn into more questions, and you'll be hearing more from us in the coming months. So I thank you so much. If you have any questions, comments, concerns, please feel free to email me. My email will be coming out after this presentation is done, and I look forward to seeing you guys and working with you in the future. Thank you.